I'm Ruth Nisley. I'm from the Animal Diagnostic Laboratory, which is a part of the Veterinary and Biomedical Sciences Department at Penn State. At the Animal Diagnostic Lab, we perform a variety of tests to monitor the prevalence of disease that is found in various livestock species. Infectious diseases that are caused by viruses or bacteria are quite important to detect at an early stage because they can spread really rapidly through herds or flocks, especially when you have livestock animals who there's a lot of them all in one place. So we're always on the lookout for new technologies that we can use to develop swift, sensitive, and specific assays. Two of the newest technologies that we have are called RealTime PCR. This technology gives us an output that looks like this graph where there's a line indicating that the thing we're looking for, the bacteria, is present. And it's testing the presence of the DNA from that organism. This is made possible through genome sequencing. So we can take genome sequencing information from a virus or a bacteria and find a small piece of that DNA which is unique to that organism, and then develop a PCR test specifically for that organism to test for that piece of DNA. Today, I'm going to tell you about two examples of how we used this next generation genome sequencing technology to develop rapid real-time PCR assays against two emerging bacterial diseases in Pennsylvania livestock. The first of these is a disease that is called Infectious Cattleya. It's caused by Avibacterium paragallinarum. And coryza is an upper respiratory disease. It affects growing broiler and layer chickens. I'm going to warn you that I have one yucky picture. It's going to show up now. It's a chicken that is showing the typical symptoms of Avibacterium paragallinarum infection. It's facial swelling, as well as nasal discharge, conjunctivitis, and the animals who are infected with this have coughing and sneezing. The chickens also have a drop in egg production, as well as a reduction in body weight or meat production because they eat less and drink less. The Avibacterium paragallinarum infection can spread very quickly through a poultry house, leading to most birds in a house showing symptoms within just a few days. The traditional methods for diagnosing coryza take several days to first isolate the bacteria and then to positively identify it as the correct species using different biochemical and protein tests. To make it even a little more complicated in this situation, Avibacterium paragallinarum is one of several different Avibacterium species that grow in chickens. And so it can be really hard to differentiate between these closely related bacteria. So that is why we rely on these additional tests. So altogether, it's going to take from 1 to 4 days to figure out what it is you're looking at if the animal presents with this type of symptoms. Beginning in early 2019, we started seeing some outbreaks of coryza in Pennsylvania chickens. And we didn't really want to wait for 4 days every time one of these things happened to find out if that's what was causing the disease. We needed a rapid and specific test. Unfortunately, there was not one available to us, largely because the genetic sequence data for Avibacterium paragallinarum was very sparse. So we needed to kind of take matters into our own hands. The bacteriology section at the Animal Diagnostic Lab partnered with our newly formed genomics core so we could actually sequence our own isolates and get that sequence data that we needed. So following the sequencing process, which takes a couple of days to go through this workflow, we performed data analysis to identify a portion of the bacterial DNA that was unique to Avibacterium paragallinarum. This allowed us to then make a real-time PCR assay. After many months of work, we ended up with a PCR test that has a limit of detection of 4 bacteria, meaning it's very, very sensitive. We also made sure that it was very specific so that when it comes up as positive for this DNA, we know that it's just this Avibacterium paragallinarum and not one of the other viruses or bacteria that can cause similar symptoms, nor one of the closely related Avibacterium species. So we validated that this was true, and the assay was quickly added to our client offerings. It still continues to be run about 2 times every week. So we got the coryza outbreaks pretty well under control. At least we can diagnose them quickly if they pop up. And of course, as soon as we started running that test, a new outbreak happened in a different species. This one has a little bit of a backstory as to how it got to Pennsylvania. So starting in early 2019, North American swine producers in Canada and then the US began noticing that there were die-offs associated with the detection of this bacterium called Streptococcus equi subspecies zooepidemicus. I'm going to call it strep zoo because it's a lot less long. And the USDA noticed that this bacteria was showing up in a lot of kind of unexplained pig deaths. So in 2019, they issued an emerging risk notice, and they began investigating the genomic structure of this bacteria. They found that the strep zoo from these outbreaks matched that from virulent strains that had previously been associated with swine infection in other countries and with human infection in the US. Virulent strep zoo infection in pigs results in sudden jumps in spontaneous abortion. A pregnant sow would lose her piglets before giving birth, and also sudden illness in adult and growing pigs. The affected animals can shift from being apparently healthy and exhibiting no symptoms to being weak, lethargic, they can't get up, and have a spike in temperature, running a fever within a couple of hours. So pigs can actually die from this within a couple of hours to a day. So a response that's quick is very important. Unfortunately, in December 2019, right around Christmas time, we detected this in an event where some pigs had died, and it was kind of unexplained. If we can diagnose it early, strep zoo can be controlled through antibiotic use or by removing the sick animals from being in contact with other animals. But again, like the Avibacterium paragallinarum, it can take several days to make this identification. Quick identification is also important because it can spread quickly through the population if it's not controlled, and that the sick animals, up to 50% of them can easily die. So to make things a little more complicated, because that's the best way, of course, this strain that seemed to be infecting these pigs was a very bad bacteria. It would show us false negative results when we ran those biochemical tests. So we could grow it, and then when we further tested it, it showed that it wasn't strep zoo. So there's something very unusual about this strain. In addition to getting these false negative results, we would also be getting false positive results because healthy pigs also have strep zoo living in them. And so you have to be very careful to determine, using those traditional techniques, if strep zoo is really what's causing the disease. So we needed a way to tell the difference between the good bacteria and the bad bacteria. We therefore turned to whole genome sequencing to see if there was something specific in the genome that could help us to identify that. And thankfully, we found that there was a specific gene called SZM, which was identical in our Pennsylvania strains to that to the same gene that had been found in virulent strains from other countries. This was an important finding because it was also not found in the good bacteria. So other strains of strepzo didn't have it. So this was exactly what we needed to identify this. We quickly, again, developed a real-time PCR assay so we can test for this in about 2 hours as opposed to 2 days. And we can detect as few as 6 bacteria. We also made sure we validated this in a lot of different types of samples. Getting a sample from a live pig who's sick is not super easy. So we wanted to make sure we could get it from lots of different places. And in addition, we made sure it was very specific to the bad version of strepzo. We made sure first that we wouldn't get false results from related viruses or related bacteria or viruses that could cause similar types of symptoms. And also that the good bacteria were not detectable while the bad bacteria were identified. So altogether, these 2 examples helped to support an industry of swine and poultry production in Pennsylvania, which is valued at over $1.8 billion annually. These quick tests that are sensitive and specific are really critical for accurately identifying the causes of infectious disease. And so I, of course, need to thank all the people who actually did this for the support of the staff of the Animal Diagnostic Lab, as well as the funders who allow us to be able to do this work. We hope to be able to continue to improve diagnostics and help keep Pennsylvania livestock industry strong and healthy. So that's the end of my story of what we've been doing the last little bit here. Be happy to take any questions if people have them.